Our third lecture in the series is going to be on the process of protein translation. For this, you should be able to explain the genetic code and what open reading frames are. In addition to that, you should be able to explain the process of translation from the beginning, from initiation to termination, and how certain drugs and poisons interfere with translation. So just to start us off, we said in the last lecture our genome contains lots of information, some that's coding and some that's non-coding. Specifically for today's lecture, we're going to be talking about those parts of the genome that code for proteins. And the complement of proteins expressed in a cell is called its proteome. Proteins are synthesized in the cytoplasm by organelles known as ribosomes, and translation often has a defining role in the control of cell behavior. So even though all cells will contain the same genetic information, only a subset of that of those genes are actually coded for in any given cell, and this is what gives cells its their unique appearance, their unique function, because of the subset of genes that are expressed in that cell compared to other cells. So dysregulation of translation has been linked to a variety of diseases, and as a result of that, translation is a target for various drugs, but also for poisons as well. A lot of what we're going to be talking about today is based on something called the triplet code, and the molecular machinery, so the molecules that are involved in translation, recognize three nucleotides, three pieces of DNA in a row called a codon. So the molecular machinery recognizes codons in messenger RNA by something called an anticodon-codon base pairing. And in order to do this, it needs molecules, RNA molecules called aminoacyl transfer RNAs, or tRNAs for short. So these molecules have what's called an anticodon, and it's complementary to the three base pairs that are on the RNA, and they bind a particular amino acid also. So on one end they have an anticodon that recognizes the three nucleotides, and at the other end of the molecule it has a specific amino acid that will only be ever linked to those three nucleotides. The code is the same for almost all organisms. In addition to that, mitochondrial messenger RNAs have some slight differences. So this is the table that has been deciphered. So on the left hand side we have the first of the triplet codes, so the first nucleotide in the triplet code. The top row is the second of that combination, and the column on the right tells you the third letter. So if you want to find out what amino acid a particular sequence of three nucleotides codes for, this is the table that you can use. So for example, if you take the amino acid phenylalanine, which is in the top left hand corner, this is coded for by the nucleotide sequence U, U, U. So U from the column on the left, U from the row on the top, and U from the column on the right. So combining these three nucleotides together will be recognized by a transfer RNA molecule that is carrying a phenylalanine, and it will always be this case when it sees the U. If we take a second example, so the amino acid serine, or SER, so that's second in from the top left, this is coded for by the nucleotide sequence U, C, and U. So U from the left hand column, C from the top row, and U from the right hand column. So anytime the transfer RNA molecule specific for serine carries this sees this UCU, it will bind to it via its anticodon and carry with it the amino acid serine. And this is how protein translation is progressed, by the transfer RNA molecules recognizing specific nucleotide sequences in combinations of three and carrying with it a specific amino acid for that triplet code. You'll notice that from the table there are a number of amino acids that appear a few times. So this gives us a certain level of redundancy, meaning that there are more than one combination of 3 nucleotides that code for that specific amino acid. You'll notice with phenylalanine, for example, you can see that there's 2 different triplet codes that can give you phenylalanine. If you look at some of the ones on the rest of the table, leucine, LEU, has 6 different combinations that give the amino acid leucine. In addition to that, as well as actual amino acids, there's also a triplet code to stop the translation process, and these are known as stop codons. There's 3 such stop codons that exist, and these are usually, as the name might suggest, at the very end of the translation process, and we'll talk about that shortly. So in order to determine what amino acid goes where, we need something called a reading frame. So if we take the same messenger RNA sequence, and I've given you an example here on the right hand side, so those 3 lines of code, those 3 lines of U's, A's, G's and C's, they're the exact same nucleotides, except they're arranged in 3 different ways. And you can see this, for example, with the top one. The first triplet code is UUA, and it's starting at position 1. In the second example, it has the codon starting in position 2, so rather than using the first U, it's using the second U, and it's coding UAU as the first amino acid. And the third possibility in this regard is by starting at the 3rd position, so skipping the first 2 U's and starting with the A, and it goes AUG. Now, only one of these reading frames is possible. It'll only produce a functional protein. Now, why would this be? Well, if we take the first one, for example, it will read the first codon and it'll give you a leucine, but by the time it gets to the second codon, it codes for a stop, and this will ultimately only end up with 1 amino acid protein, which isn't viable. It's not going to be a functional protein. The same situation is true of the second example, where you have 3 amino acids that are able to be synthesized, but then it's followed by a stop. So this 3 amino acid peptide will not be functional. The third option, though, does give you a functional protein, and usually the correct reading frame is set in vivo by the start codon AUG, and this always codes for the amino acid methionine. So quite a lot, if not most, proteins that are coded or translated begin with the amino acid methionine, MET. Now, this might not always be present because, as we'll talk about later, there are post-translational modifications where it might get cleaved or modified in some way. But initially, when it's translated, most proteins will begin with the codon AUG, therefore it'll translate into a methionine. So by definition, an open reading frame, or an ORF as it's called for short, is a run of codons that starts with an AUG or an ATG and ends with a stop codon. When the protein is known, the open reading frame is called a coding region. When it's not known or hasn't been discovered, it's simply just called an open reading frame. In terms of the directionality, we were talking about this the last 2 lectures with regards to transcription and for DNA replication. When mRNA has moved from the nucleus and has moved into the cytoplasm to the ribosomes, it is read from a 5' to 3' direction, meaning the 5' carbon at the uppermost and the 3' carbon at the end. This leads to the translation of a protein in the N to C terminal. So N stands for the N-terminus, which is normally denoted by an NH3 or an amino group, as indicated in the diagram, and the C is where the C-terminus is, or where is very often a COOH or COO group, a carboxylic group. So most amino acids will have this layout where you have an N to C terminal in the protein direction. In addition to that, there's also what's called a peptide bond, which is highlighted in grey in the diagram, and this is always a C double bond O combined with an NH. So the C double bond O comes from one amino acid, the NH comes from the second amino acid, and between them, when they bond together to form a chain, it's called a peptide bond. So just very, very briefly to go back to the transfer RNA molecule, which we mentioned at the start. This transfer RNA is a different type of RNA. It's not messenger RNA. It's RNA that's present at the ribosome, and it contains usually 76 bases made up of A's, U's, C's, and G's, and some other modified nucleotides. And in its three-dimensional structure, it has a cloverleaf-like structure. So this is shown in the diagram on the right, where it has many loops, 3 major loops that exist. In particular, I'd like to get your attention to the anticodon loop in particular, because this is the part that actually binds and recognizes the nucleotides on mRNA. You should also see that at the very, very top, there's an amino acid attached, and that's the amino acid that corresponds to the codon, or the 3 nucleotides on the mRNA that is specific for that particular amino acid. So this means that there's actually amino acids, that there's transfer RNAs for all the amino acids. So the amino acid is covalently bonded on the 3' end of the transfer RNA. In addition to that, the amino acid is attached or bonded by an enzyme called aminoacyl transfer RNA synthetase. And as I said already, there's at least one transfer RNA molecule for each amino acid, so there's 20 in total. And as we saw already, some amino acids have more than one. So the transfer RNA molecule itself is synthesized by RNA polymerase, and not RNA polymerase 2, as we mentioned in the last lecture, but RNA polymerase 3. And it's typically synthesized by as larger precursors. So all transfer RNAs are subject to a variety of chemical modifications before they leave the nucleus. In fact, nearly 1 in 10 nucleotides are actually altered. There's over 50 modifications known, and these can be as simple as things like methylation, where a CH3 group will be added to the nucleotide. These are modified because they affect the conformation and the base pairing of the anticodon in order to facilitate the recognition of the appropriate mRNA by the transfer RNA. So by modifying all these different nucleotides on the transfer RNA molecule, it gives them a unique shape that's specific for the nucleotide sequence that it needs to recognize, but also for the pairing of the amino acid to its 3' end. So the linking of the amino acids to the transfer RNA requires energy, and if you remember a few moments ago I said that the enzyme involved was aminoacyl transfer RNA synthetase, and this is shown in the diagram here at the very, very bottom. So in the very bottom left you can see the transfer RNA synthetase, and in this case it's specific for phenylalanine, so it will in one binding cleft bind to phenylalanine, the amino acid. In the other binding cleft it will recognize the transfer RNA specific for the phenylalanine. It'll use ATP then to link the two together, so it'll chemically bind the aminoacyl transfer RNA with the amino acid, and the net result is that you have a transfer RNA linked to a specific amino acid that is only recognized by a particular triplet codon. So this ensures that the right amino acid is always linked to the exact correct transfer RNA molecule. So the correct amino acid has the highest affinity for the active site of that particular synthase and is favoured over all of the others. So as mentioned already, the ribosomes is where the translation actually takes place, and unlike transcription and unlike replication, there are quite a few mistakes made in this process. So it's estimated that there's a mistake made in 1 in every 10,000 amino acids, which is much, much higher risk when you compare it to something like replication, where it's 1 in every 10 to the 9 nucleotides where there's a mistake. So there's estimated to be over 1 million ribosomes per eukaryotic cell. The ribosomes contain another type of RNA called ribosomal RNA, as well as specific proteins. Initially there's 2 subunits that separate when not synthesizing, so this means that you only get synthesis of new protein when these subunits are joined together. And they join together on a messenger RNA molecule near the 5' end. So it takes that arrival of a messenger RNA molecule for the two molecules for the two subunits to actually join together. And when a stop codon is reached, they separate it. It's estimated they can add around 2 amino acids per second to a polypeptide chain, and the two subunits are called 60S and 40S, and joined together they form what's known as an 80S ribosome. Now, you'll notice that 60 and 40S do not give 100 because this is not specifically additive. It's not referring to their molecular weight. It's actually referring to something called the sedimentation coefficient, or Svedberg unit, which is what the S stands for. So in terms of how translation begins, the initiation procedure begins with the cap structure. So we mentioned this briefly at the end of the last lecture. And the 5' cap, which is purposely added onto the mRNA molecule, enhances the translation of mRNA by helping it to bind the eukaryotic ribosome and ensuring that there's recognition of the proper AUG, or methionine initiator codon. In order to do this, it needs a consensus sequence, and very commonly this will be something called a Kozak sequence. So this is shown in the sequence of letters in this particular slide, and this is considered to be the strongest ribosomal binding signal in eukaryotic mRNA. So for efficient translation initiation, the key elements that need to be present are a G or guanine residue at the first position and an A residue at the -3 position. An mRNA that lacks the Kozak sequence may be translated efficiently in cell-free systems, but it lacks a stable secondary structure. So the ribosome will bind to a particular purine-rich sequence or region called the Shine-Dalgarno sequence, which is upstream of the AUG, or the start point. And this sequence is complementary to the 3' end of the 30S ribosomal subunit. And the position of the ribosome binding site in relation to the AUG is very important for efficiency of translation, and it's normally either 6 to 10 spaces away from the A of the initiation site of the AUG. So this is just showing what happens during translation. The very, very first amino acid that's made. So for each ribosome there'll be 3 binding sites for transfer RNA and 1 for mRNA. The A site binds to an incoming aminoacyl transfer RNA. The P site binds to the transfer RNA linked to the growing polypeptide chain. And the E site, or the exit site, binds the transfer RNA prior to its release from the ribosome. So you'll notice that the first amino acid here, or 1 in the diagram, binds by the anticodon to the mRNA, and it has an amino acid. In this case it's methionine, indicated by 1, and that is brought in at the very, very first position of the ribosome. It'll then subsequently have other amino acids and other transfer RNA molecules join it, and they can be now chemically bonded to each other to form a polypeptide chain that then gets bigger and bigger. So this is just showing what happens to the actual ribosomes themselves. As mentioned, you have 2 separate subunits, a 60S and a 40S, that combine together to give you an 80S. But in order for this to happen, it needs several other proteins to form it, and this is known as an initiation complex. So just the diagram on the right shows you all of the other proteins that are needed to combine. The first thing obviously is the recognition of the cap, and this is indicated in the diagram by the M7G cap, and a number of other initiation factors, elongation initiation factors, that have to join and bind the ribosome in order to get the second subunit to actually come along. So it is a carefully sequenced order of events, and these all need to happen in the right order for the ribosome to form with the mRNA also bound at this particular position. So the elongation process simply refers to the growing of the polypeptide chain. So you can see in the diagram on the right that there are a number of transfer RNA molecules arriving from the right, and each of these have their own unique anticodon that then recognizes a codon on the mRNA molecule in red. They bind, they arrive, they transfer their amino acids that they're carrying to the growing polypeptide chain, and these are chemically bonded by a peptide bond. And once they move along the different positions of the ribosome, the A, the P, and then the E site, they then leave, and you can see this on the left of the diagram where a tRNA molecule is leaving without its amino acid. This will now return to be joined with another amino acid that it is specific for. So this means that the ribosome is moving along the mRNA molecule, reading it in codons, groups of 3 nucleotides, allowing the binding of a transfer RNA molecule specific for that codon. It arrives with its specific amino acid, it chemically bonds to the polypeptide chain, and then it finally leaves. So this is a 3-step elongation cycle, and it involves translocation from one position to the next. So 3 elongation factors are required for this, and this will not happen unless they are present. So the ribosome moves from 5' end to the 3' end of the mRNA molecule being translated, and it requires energy. So this is takes the form of GTP, and this GTP causes ribosomal conformational change. It also allows for the formation of peptide bonds which are needed to join the amino acids that arrive to the growing polypeptide chain. Energy is also needed for the ribosome to translocate, to move from the 5' end to the 3' end of the mRNA molecule and move along each of the 3 nucleotide sequences at a time. Finally, the process of translation will stop once a stop codon is reached in the arrival or the A site. Because the transfer RNA molecule that recognizes this anticodon will not have any amino acid attached to it, because it's coding for stop, it needs to have something else, and these are eukaryotic release factors. So this causes something called peptidyl transferase to release the polypeptide, and as a result the ribosome dissociates from itself. So this is shown in the diagram on the right, where you have a transfer RNA molecule, a codon coding for stop, that's recognized by the eukaryotic release factors. There's no amino acid joining the polypeptide chain. It causes the peptidyl transferase to cleave and the last amino acid off, and it allows for release of the polypeptide, and this also encourages the ribosome to dissociate from itself, all the elongation factors to dissociate from the complex, and for the mRNA as well to leave. And you end up with the 60S and the 40S subunits again. So this process is essential for cell survival, not just for human cells but for all animals and eukaryotic cells. So it is essential that the process occurs without any glitches. Some poisons have been known to damage or inhibit the process of translation, and one of the most toxic substances known, called ricin, is such a substance that actually damages or targets the ribosome. So ricin is a toxin composed of an A and a B chain. It is found in the castor oil plant, and in particular the ricin toxin itself is found in the beans, and the diagram on the right shows what the beans actually look like, and also the plant. And it essentially can bind to the ribosome and can affect the ribosomal RNA. So if it affects the ribosomal RNA, you can't get proper assembly of the ribosome, you can't get proper translocation along the mRNA molecule, and as a result you can't get the creation of new protein. So if our cells can't make protein, it means that they won't be able to survive or carry out their regular functions. Very, very acute effects include things like vomiting blood, diarrhea, renal and respiratory failure, and eventual death within 14 days, depending on the dosage. So this is a very, very toxic substance, particularly when you compare it to substances like cyanide, for example. However, targeting the ribosome can be beneficial in some instances, in particular for targeting bacteria and killing bacteria. They too also need an efficient ribosome to translate their protein, particularly if they're dividing very, very quickly. They need to make a lot of new protein. So some of the classes of antibiotics in particular target the ribosome. And gentamycin is one such example. It's bactericidal, meaning it kills bacteria instead of stopping its growth, and it binds to the 30S ribosomal unit of bacteria and induces misreading of the mRNA during the elongation phase, leading to incorrect synthesis of proteins, and it means that the bacterial cells can no longer survive. So in summary, the genetic code is a triplet code that determines amino acid composition of peptides. The process of translation is regulated at the initiation phase, the elongation phase, and finally at termination. And there are a number of drugs and poisons that can interfere with the process of translation.